why. Who's been using this?
Duncan.
Duncan. Mr. Duncan.
All right, the first thing about this thing is it's huge, right? And you can't fit it all in one image. This picture that you have here is from a dissecting microscope, right? So you can't fit it all in one image. Then secondly, the colors are weird, right? Like this one is purple, and that one is silver stains, right? Or what are called metal stains. And then you can have red ones, you can have purple ones, you can have, I've seen green ones before. You can stain it almost any color. But the thing about the central spinal cord is that you have to worry about the two parts of it, right? And some are easier to see than others. How's this one for a comparison between the two parts? Can you see them fairly easily? I can, I don't know if you can, right? Can you see the two parts or not? You got this part over here and then this part over here, right? They look different. See, here's the defining line, right? So you can see it, I don't know what you call it, structural difference. How about that? No? It all looks the same to you, right? Okay. So, let me see if I have another spinal cord one. For some reason, that's the only one they have in this box. Time to clean my desk that's been used by too many professors. What?
At the risk of sounding
Stupid?
Yes. I don't think that you posted the central nervous system lab to the grad student.
Yeah, I don't think you did either.
And
The risk of sounding.
I'm having him email it to me though.
do you want this one?
I think we have one being airdropped, but I would take that if.
Being airdropped?
Yeah.
You need a parachute?
Yeah.
Some kind of technology I don't know how to do.
Yeah, it's like possibly.
So slide boxes behind you. See if you can see one that has central spinal cord slides in it. Because the one only one I have is the red, the pink, and that one, whichever the one I just showed you, and it's not the best. Oh, you know what I could do? Use that one brain cell I got. Make it work.
Hey Matt, can you email it to me?
Sure. Sure, no.
LUKMWOO
KMW.
at email or at ecok.edu.
Isn't it email that easier?
No, I think it's because I'm faculty now or something, staff. I think
No?
it will work with email though.
Maybe I'll try that. Not gonna be junk, obviously. You're gonna right here.
There's a major search going on back there. Look at that. It's like the joke about how many people does it take to screw in a light bulb.
And then try your junk.
Well, no. You're saying it's not there.
Might be, I mean, there's like.
Hey Kaden. Kaden, I
Look at
sent
some
you
other
an email.
Hmm?
I sent you an email, maybe.
You sent me an
Oh,
email.
I got it.
You got it?
Yes,
Yeah, we
thank
got
you.
Okay.
Oh,
Oh, they're
sorry,
just
no,
looking for
sorry.
slides, aren't they? So they're looking for paper.
I could not get it. I would try to answer you already. I'm just in like my good notes.
Oh, I appreciate it. Appreciate the effort.
All right.
Sir,
No, right. Well,
I just did a bunch for the summary. It was like a summary and critique. Like I'll just summarize all this and like my critique. It can be positive and negative. The one thing I was like, I wish I had like a graph or something for like for the total population. Cause like we chose like this 1,000 people from these years.
Yeah.
That is okay. That's like.
A graph of, like West Virginia with dots and
all right, let's go to a different color. Here's one of the other colors. Let's look at this one, see how this one looks. Is better or worse?
That's better. Better.
Because there's a color difference. Yeah, okay. All right, we'll start looking at this, see what it does. All right, so. Oh, this is a weird one, okay. It's on there kind of sideways. So top and bottom are this way on this one. All right. Let's start with the easy stuff, right? White and gray matter. Ooh, let's fill that one. Jinky. Jumping back. White and gray matter, where is it? No idea.
Your matters in the interior, then
The first lab we looked at one of these things, by the way, I don't know if you know that. The very first lab we looked at this slide. Nobody remembers that. Nissel bodies, yes.
Oh.
The Nissel bodies are, let's see if I can find a good spot where you can see them here. You can see them here. Let's go over here. These are better. All right. Nissel bodies. In these motor neurons here. Right? So, what's the difference between white matter and gray matter? Who am I going to pick on today? Who got my high score on the exam? Or maybe who got my low score on the exam? What are you gnawing at? How's that working for you?
I think I'm working out my jaw, like, muscles.
Gotta get those jaw muscles up. Nobody wants to talk today? Is it like fall break already? We don't care? I know that's next week. You don't have lab next Thursday, so you're just taking the vacation early.
I'm gonna guess that the outer part is the gray matter and the inner part is the white.
What's the difference?
opposite.
Remember the difference? I talked about it yesterday in lecture, right?
gray matter does like a stone body.
The gray matter is called gray matter because it does not have myelin. The white matter is called white matter because it does have myelin, right? So you're not going to have nerve cell bodies like motor neurons in white matter. You are going to have it in gray matter. So what did I just point out to you? What are these? Motor neurons, right? So where is that? Gray matter. That means that this is the white matter, right? On the spinal cord, the white matter is on the outside, the gray matter is on the inside.
Brain is off.
In the middle, so there's two lobes, right? In the middle, there's this canal, right? Central canal. And then the two lobes are connected by this central passageway that goes where the central canal is, called the what? Commissure. This is the commissure, that gray matter connection between the two halves. The central canal on this one looks like it's full, right? They don't all look that way. Let me look at a different one. Show you a different version of it. On this one, notice that the central canal looks full too, right? See, it looks full there too, right? But if I go to a different one, let's see. Let's try this one. Yeah, look at this one. How's that central canal look? Very different, right? So the cells, this is a really nice one. It's really nice and clear. You can see all the structures in it. It's really nice. Nice, right? So the cells that line the central canal are called that E word there, which is another one of those words that people say differently, right? Some people say ependymal, some people say ependymal. I don't care how you say it, spell it, right? There's an old adage, right? Don't make fun of somebody when they mispronounce words because they learned it by reading it. Never complain about somebody actually reading something, right? Okay, where are we? Have we done everything? Oh, posterior and anterior horns, right? That's always the problem area, right? What's the posterior horn? What's the anterior horn? Somebody look it up and tell me.
The one with the,
The other problem, go ahead.
oh, I just try to look for the dorsal root ganglion on it.
That's not always the case though. The dorsal root is, that's another whole story all by itself. So there's three different stories here that are a problem and they're all linked together, right? The first one is that the name is weird, right? There's two different names for it, right? There's dorsal and posterior and anterior and ventral, right? Which don't mean the same things at all, right? On us they do, but on other organisms they don't, right? So, on us, the dorsal and the posterior are the same, and the ventral and the anterior are the same, right? And you'll see it listed both ways. You never know how it's going to be portrayed, right? That's one problem. The second is on this picture, you can see the dorsal root ganglia is kind of down toward the bottom, right? Those things sticking off to the side like wings over there, right? So is that the dorsal side or is that the ventral side? Good question, right?
Just extend it up, I think it's the way it's cut. I think the top side is the dorsal side.
And then the last part, let me go down to my lowest power again. The last part is if you look at this one, everybody can tell where the gray and the white matter is again, right? You can see that this horn here comes down and kind of rounds off down here, right? On the other end, let me go up. On the other end, it kind of comes to a point, right? It comes to a point, and actually in most of them it goes all the way to the edge. So one of them pretty much connects out to the edge and the other one doesn't really. Although it does, which is kind of weird, right? That's another whole story, right? So now we have to figure out which one's anterior, which one's posterior, which one's dorsal, which one's ventral, right? Are you totally confused now? So what's it say in your books that you have running around, or your internet searches that you have going on? No? I used to have a big poster in here, but I took it down because people were cheating on it.
posterior is gonna be at the top part in this diagram.
Say that again.
The posterior is gonna be at the top one on this diagram.
So is the posterior part, they're called horns, right? Of the gray matter, the horns. You get horns on each side, each end. So the simple answer is the dorsal horn the pointy one or the rounded one? That's the question, right?
Pointy one.
The pointy one is the dorsal or the posterior horns. Okay? That means this one is dorsal, which means on your body, where does it sit?
In the back.
Toward the back, right? So notice where the dorsal root ganglia are though. They're on the ventral side. Isn't that funny?
Is that always the case, or is that just?
Pretty much.
It looks like it extends more from the dorsal, like it looks more connected to the dorsal side.
It is more connected because there's a bigger bundle coming there.
Yeah.
Now, who can tell me which, so the dorsal or the ventral, right? Extensions, which one is the receiving, which one is the sending again?
I agree with that.
He says the sending is dorsal and the receiving is ventral, right? That's what you said? Everybody agree with that? Sure, whatever he says, right? Okay. Any questions on that?
Do you agree with it?
What?
Do you agree with it?
Yeah.
Okay, so not sure.
All right. We want to go to the next page? Well, that's the central canal and the commissure and all that stuff, right? Gray matter. Let's look at the motor neurons. Yay, motor neurons. These are nice motor neurons here. I like them. They're not as good as the other one. Let's look at the other one. This one. These motor neurons are killer, right? How do those look? Pretty good, right? Motor neurons in the gray matter, right? Primarily located in the, which horn? Ventral horn, right? The rounded one, right? So they're in the gray matter, they're motor neurons. I don't know what else I can say about them. I mean, you can see them, the nuclei is blatantly obvious, right? So what's the nucleus here? Is it the dark thing or the little white ring around it?
White ring.
It's a little white ring around it, right? What's the dark center? Nucleoli. Anybody know what a nucleus that has a centrally located nucleoli is called? There's a name for it. What?
Repeat 4 for 4.
Repeat 4 for, what do you want me to say it again?
The whole thing.
What's the name for a nucleus that has a centrally located nucleolus? You get it that time? No? It's called a vesiculated, vesiculated nucleus. I won't test you on that. Just for your erudication, right? And what's the other thing you see in here in these guys? First lab, Nissl bodies. See the grainy looking stuff there in the cytoplasm? Nissl bodies. Anything else there? No, that's all it. That's it. Pretty simple. The white matter. Yeah, the white matter's fun. We got all kinds of stuff in the white matter. So the first thing What are you talking about? All right. This is my lowest power again. Unfortunately, it's my lowest power. I can't go lower. Sometimes I wish I could, but I can't. So what is the gray matter? What does it look like if you look at it from a high magnification? If it looks right. None of these are good. If you get a good looking one, what does the gray matter look like if you look at it, kind of stand back from it and look at it? Say it again. Butterfly. Okay. So, what am I looking for? Oh, so above and below the central part, right, there's a central canal with the commissioner going across, right? If you look above and below, you can see the two halves of it, right? So here's the division between the two halves, right? Everybody see that? So which horn is that on?
The neutral.
This is the rounded one, so which way is that? Anterior, right? So this is the anterior, what do you want to call it? It's on, we're on number 3 here, this one.
Median fissure.
What do you think it is? Anterior median fissure, right? In some cases, that's really big, by the way. This anterior median fissure can be really wide, depending on where it is in the spinal cord. So what's the other side called? If you go on the other side of the commissioner, up here. This thing here. Posterior median sulcus. What's the difference between the posterior median sulcus and the posterior median septum? What's a septum? Wall, divider. So that's going to be this dark connective tissue line that goes up there, right? All right. The labeling's all messed up on that. I don't know why. It's been that way for a while and I have never taken care of it. All right.
Wait, so the
The
posterior,
the lines on this don't matter. You make your own lines.
the posterior median sulcus is the space.
Sulcus is actually the sulcus, if you really want to be. Sulcus is this little triangle thing there.
Oh.
Oh, wait, I think I see it, okay.
What am I looking at next? What am I missing? Neuroglial cells. What's a neuroglial cell look like? And where are they going to be? That is some weird white matter there, man. Let's look at this one. So we're going to look at the white matter. We're going to zoom in on the white matter. This stuff, right? This is white matter at a high mag, okay? And what's it look like?
Looks like lymphocytes.
Kind of weird, doesn't it? A really good image on it is this one. This is a really good cut here. Just happened to catch it just right? What is that?
Oh, is that the axon?
So the white here is the myelin sheath around the axon, right? So there's a nerve fiber, if you will, but it's not a nucleus, right? It kind of looks like a nucleus, but it's not, right? The only nuclei you're going to have out here in the white matter is what?
Neuroglia.
Neuroglial cells, right? You may have some what? The ones that lay down the myelin.
Oligodendrocytes.
Oligodendrocyte cells, maybe, right? So where are those nuclei at?
The really dark spots.
Like here, and there, and there, and there, right? Those are little microglial cell nuclei. Anything else? Pretty easy. You don't really have a good picture of it, but that's what it is. Probably should take some notes. All right, now we're gonna look at the meninges, right? One of those favorite words, right? A meninge. Let's look at this slide. This one has really good meninges, so let's look at the meninges. Let's look down here. This looks very much like the picture. It's not quite the picture, but it's close. Right? Isn't that close to the picture? Not quite the same, but almost. There's too many big blood vessels in there, right? They don't match up. So it's not the exact same slice, but it's the same stain, same area, lots of different things there that are very similar. Am I boring you?
No, I just had to be on.
Late night last night?
What? Wait, what?
I said, did you have a late night last night?
Yeah.
Those video games, I'm telling you.
Yeah, Fortnite Lego.
All right. What's this?
The dura mater.
Dura mater, right? And what's this area here?
Arachnoid layer.
Arachnoid, right? And this kind of purplish little band here is the pia mater. These are what?
Blood vessels.
Blood vessels, yes. And then the other thing it asks for is capillaries, right? The pia mater capillaries are really small. You have to really hunt for them and really dig for them a little bit. But I'll show you. We'll find one down here. As I said, they're really small, but they're important, and that's why we're looking at them at all. There's one there. Everybody see it? It's really huge, right? No, you don't see it? Right there? Tiny, right? They're not big at all. All right. What else? Anything else on the spinal cord? No. Let's go on to the next one. So that's it for spinal cord. Let's go on to something else. Multipolar neuron slides. The slide we're going to look at is nerve cells, isolated spinal cord smear. Anybody know what that means in English?
The juices of the spinal cord that's just been.
So they basically took this like a section of a spinal cord and put it in some chemicals so it would break into pieces, and then just smeared it on the slide, right? So you can get isolated single cells out here that are all alone, so you can see them easily, like this guy. Everybody see that guy there? Thing looks like a weird tribal mark up there. Look better now? I'm still not seeing what I'm looking. I can see it better on my scope than you can see it. You can barely see that nucleolus in there. See if I can find a better one. That was a good one though, I thought. I have to get one of these dark stained ones. about that one? How's that one look? Better? Darker? See the nucleolus? Yes, you can see it. Okay. All right. Multipolar neuron, right? What's first? Nucleus. Oh, okay. Where's the nucleus? Nucleolus right in the middle, right? What's the Nissl bodies? All this dark stuff, right? Which one's the axon? Can you tell?
No, no say.
We would assume that this is the axon, but we don't know that for sure, right? So when in doubt, what do you do? They're all axons, right? We know that there's one axon and multiple dendrites, so we can make some guesses, but when in doubt, they're all axons, right? Okay, anything on that one? Pretty simple, right? Straightforward, nothing hard. Okay. Next, we're gonna look at the brain. Here's the fun part, right? There's two parts to the brain. Everybody knows this, right? Technically, there's 4 parts to the brain, right? Would you agree with that statement?
Yes. I don't know.
How do I mean?
So there's like
Yeah.
a little different.
There's 4 parts, technically. Yeah. There's the cerebrum and the cerebellum, right? Everybody agree with that? And each one of those has 2 halves, right? The models, right? Think about the models in anatomy lab. You pull them apart, right?
You're talking about the
Do
ones
that.
like the parietal and occipital.
Oh, we haven't done those yet. That's a whole other thing. So which one's part? What are the 2 parts and where are they? In your head. What's this part here?
Cerebrum.
I know they're very similar in spelling and pronunciation, right? Cerebrum and cerebellum sound very similar. So let's make them as confusing as possible, right? But what's this one on top called? And this one back here? And which one is the tree of life?
Cerebellum.
Cerebellum, why? What's it do?
Autonomic stuff.
Eyesight.
Has
It's
all
the one that
the-
keeps your heart beating, right?
Yeah.
It does all the maintenance stuff, right? Cerebrum does what? What's this part do here? This is your thinking spot, right? You think up here, this is your mechanical stuff back here, right? Think free-form thought and mechanical upkeep, right? Piece of cake. That's all you need to know, right? Oh, wait a minute, there might be more. Which one first? What are we looking at first?
Cerebellum.
Is that what we're looking at? Do I want to look at the cerebellum first?
Yes.
So the cerebellum can be same as the others. It can be a non-metallic stain or a metallic stain, right? So it's going to look either purple, like this one, purple and pink, right? Or it's going to be that silver metallic stains over there. And they are good at showing you different aspects of this tissue. So here's another part that's always fun, right? Is that just like everything else, this has gray matter and white matter just like the rest of the spinal cord, but instead of calling it gray matter and white matter, what do we call it here? Cortex and medulla, like the organs, right? So again, let's confuse you some more, right? Let's make it complex when it could be simple. So where, on the brain, where's the gray matter and where's the white matter? On the spinal cord, the white matter's on the outside. On the brain, where's the white matter? It's on the middle. It's reversed, right? That makes no sense, right? Remember I said that your spinal cord smishes up into your skull when you're developing, right? So one of the theories is that it also causes it to turn inside out. And being a professor of embryology, there's evidence both ways there. Anyway,
Is it advantageous for one, is one more advantageous than the other?
as far as what?
The white matter being on the inside versus the outside?
Well, remember that, so the white matter is where the myelinated fibers are, right? And that's where you've got, think like phone cables, right? That's what that is, right? The white matter is your phone cables that carry the information, right? You cut your phone cable, you don't have any communication, right? That's why if you cut your spinal cord, you're in trouble, right? So having it on the inside of the brain is probably good because it protects it, but that means the working parts of the brain are on the outside, right? That's not so good, right? So again, it's bad either way it goes, right?
That's what I was just wondering where we.
All right, so cortex and medulla, right? Instead of white matter and gray matter, but it also is white matter and gray matter, right? So we talked about white matter, we talked about gray matter, we said that gray matter is where the cells are, right? So on this one, your cells are all on the outside. So the cortex and the medulla, cortex on the outside, remember, right? So the cortex, and remember it's highly folded, right? So this is the outside here. Let me go up to the edge, hold on. There's the surface, there we go. So there's the outside, right? Everybody would agree that's the outside of the brain, right? And this is what again? Cerebellum, back here, right? Okay, so this is gonna be cortex, right? Does that make this medulla? Or does it make this medulla? Whoops. I know, tricky question. This pink area here with no cells in it is your medulla. From here all the way out is cortex. The cortex in the cerebellum has 3 different layers. Sublayers, if you will, right? I'll bet you can't guess the names of those 3 sublayers of the cortex. It might be number 2, 3, and 4 on your page there.
Molecular, Purkinje cell, and granular.
So let's look at this. I'm going to zoom in on this layer here and we'll look at it a little bit. No, okay, all right. Is that all on there? Not really. It barely fits on. Okay, so here's the edge here, and there's the medulla there. So can you see 3 layers? You can definitely see 2, right? Everybody agree you can see 2 layers, right? You got this layer out here and this layer here, right? What are these big whopping circles here?
Purkinje cells.
Those are Purkinje cells, right? That must be the Purkinje cell layer. So you got this one, and this one, right? All right, let's talk about the names, molecular and granular. Molecular commonly is a term used for something that looks relatively smooth. Granular, on the other hand, looks grainy, right? So which one of these 3 layers looks smooth to you? This one. That means that this one must be the granular layer with the Purkinje cell layer in between, right? And then it says protoplasmic astrocyte, fibrous astrocyte, microglia, right? Can you see any of those? Not really, right? because this stain doesn't show them one. The metal stains show them one. So let's look at a metal stain.
What was this stain called?
What's what?
What was that stain called?
That's an H&E stain.
That was just H&E.
Standard H&E. This is a metal stain here. How's this one look?
Yellow.
Yellow. Yes, it does. Let's look over here. Okay. I don't really care where we're looking. Right on the edge is fine, so you can see it all. You can see the outside going in, right? So this is your medulla here, and this is all cortex there, right? And you got your 3 layers. How do your 3 layers look on this one? Are they better or worse? Well, I'll tell you, they're definitely different, right? So there's your 3 layers. So what's this layer out here? What's this layer here?
Granular.
And what's this layer here?
Energy.
Easy, right? So the other thing we gotta look at is astrocytes and microglia, what they are. So a metal stain, where have we seen them before? Primarily, what are we looking at? Nerve fibers. They're really good at staining nerve fibers. So what do you think these little stringy looking things are?
Nerve fibers.
Nerve fibers of some sort, right? Remember what an astrocyte is, right? Astrocyte is a nerve cell with these little extensions sticking out, right? So what do you think all these weird looking little star looking things are?
Astrocytes?
Astrocytes. So that's what the metal stains are good for, showing you the astrocytes. Notice what you see out here in the molecular layer. How many astrocytes? None, right? How about in the Purkinje cell layer? Not really, right? So they're primarily where? Granular layer, right? What about in the medulla? Are there any in there? The medulla's right there, right? Right here. Yeah, lots of them, right? So there's astrocytes in the granular layer of the cortex and in the medulla. Remember in lecture I said which one's where, right? I said these occur in this part and these occur in that part. Fibrous and protoplasmic, right? Remember that? No, you weren't listening. You were sleeping, looking at your clocks, wondering when class is going to get over. No?
Fibrous is
Let's
in
do
the
this
white
then.
matter.
Let me zoom in a little bit more. You were speaking, go ahead.
Fibrous is in the white matter. So
All right, so here's some astrocytes. How do they look? Or can you even tell where an astrocyte is? Or it just looks like a bunch of weird lines, right? If you kind of step back from it and take a look at it, what does it look like? Don't do the, what's that weird little picture where you have to like unfocus your eyes and look? I could never figure those out because I can't do that. But if you kind of step back and look at the overall thing of it, you kind of almost see a pattern, right? Can you see a pattern there? Almost like this weird fishnet kind of pattern, right? Considering what they do, that's not far off the mark, right? They give you your structure, right? And in order to make a nice solid structure without being too confining, a fishnet's a good way to do it, right? But you can see them fairly well over here, right? See this kind of weird box-shaped pattern that you're seeing of these fibers, right? They make this weird little pattern with them. And that's for them making the meshwork to hold this organ together, right? So those look kind of long and stringy if you look at them, right? Here's one here. See this? There's a nuclei, and you can see like this almost an X shape from it, right? If you look closely. And then, but you can actually see the nucleus there. There's nucleus here right next to it, almost an X shape on that one as well. But if you look here, what do they look like? almost like a little round ball of fuzz, right? So, what do you think these are called? Are they fibrous or protoplasmic? Fibrous, right? They're like fibers, right? This is protoplasmic. Protoplasmic means like ball shaped, right? Microglia, different animal altogether. Microglia, you're gonna have a hard time seeing a microglia. Because a microglia is just a tiny little strip of a piece of black stuff that you're gonna see in there. And you would have to be at a high enough magnification that you could tell that it's not connected to anything else. So good luck. It's not easy to see. So don't worry about it for the test, and just remember that what its job is, right? Which is what?
It's the janitor of the.
It's the what?
It's the janitor's claims.
That's a good name for it. I like that. Janitor. So it's correlated to what?
Immune.
Macrophage, right?
Oh,
Is it just in the cortex or
They're going to be in both.
no?
All right. Any questions on the cerebellum? Cerebellum is relatively easy. I'm trying to see if I have a good image of. If you look at this slide, you see that? See that? Wonder where they get the tree of life thing, right? Hard to see any other way though. All right, enough of that. Now, that was the easy one, now to the hard one. What's a hard one? Cerebrum. All right. A couple of different stains on the cerebrum as well. Here's one. Pretty simplistic looking, right? No? Again, cortex medulla. So there's the edge up there. Where's the medulla? Right here. This is cortex as well. Folded over, right? So the cortex goes from here all the way up to there. How many layers do you think are in the cortex?
3.
4.
Does it say somewhere?
3 layers.
There are 6 layers in the cortex of the cerebrum, not including medulla. 6 layers of the cortex itself. Piece of cake, right? Everybody got them? I don't need to talk about them. All right, I'm going to come back to this slide, don't worry. Let me show you a different version of it. You'll like this one. How's this one look? You don't like that one? What's wrong with that one? Everybody see it? There's the edge right there. Everybody see the edge?
Oh, right, of course. Very.
Pretty good? You want me to put that on the test? Let me put a different one up there. See if you like a different one better. How about this one? How's this one look? Think so? Can you even tell where you are?
Nope.
How about now?
There we go.
Well, wait a minute. This looks square. There's something wrong here.
It is square.
It is, it's a big chunk of meat. Brain meat, yeah. So where's the edge? Where's the actual edge?
Bottom.
Right there. That's the actual edge, real edge. They didn't cut that part. That's natural. How'd I know? How many years I've been doing this? Too many. Can I retire? I was complaining about retiring all morning this morning. Maybe I should time that I should retire. All right. Cerebrum. It's probably the hardest thing you're gonna look at today, which is scary considering how much stuff you looked at today, right? We only looked at 3 organs and look how messy they are, right? How's this look? Pretty though, right?
Pretty is a word.
Is that cool looking? I like the look of it. I know you don't like the look of it. Let's see, where do we wanna look? Well, I could look at any of these and tell you what's going on. Do you like this lighter color or you want it darker? Like that. How about that? We'll just do it right there. That's fine. So there's the edge, right? I can't go too high on this, but we will zoom in on certain layers, right? So how many layers are there again? 6. So we're gonna start at the outside, work our way in. Number 1, this one, that says, starts with number 1, right? That starts at the out, notice it says cortex and then it says molecular layer. That's starting at the outside. So we're gonna start with the outside, work our way in. Outside layer. What did I say molecular means? Smooth looking. Do you see a smooth outer edge there?
Yes.
Up here, this smooth layer, right? That's your molecular layer, right? What's next? Outer grainy layer, right? Here, see this? This is the outer grainy layer. What's next? Starts with a P, right? Pyramidal cell layer. That's the plural of pyramid, by the way. Never would have guessed it, would I? I know, it's weird. It's a weird pronunciation. You can say pyramidal if you like. It's technically pronounced pyramidal, but don't ask me why. What's the other word that they do like that? There's another word they do that. Like the plural is pronounced totally different than the singular one. It doesn't make any sense to me when they do that.
Mouse and mice.
I don't mean that at all. All right. Pyramidal cell layer, pyramidal cell layer. So what do you think the pyramidal cell layer is going to look like? If we zoom in on it, what does a pyramidal cell layer look like?
Pyramid.
What shape do you think the cells are going to have? If you're lucky, right? What are they?
Pyramidal.
You would assume that they're pyramid shaped, right? So do we see any pyramid shaped cells? No? Yes, maybe? Let's go somewhere else, see what we see. This is not pretty. Why is this ugly? You're supposed to be kind to students. That's a good one there, but I just want more than one. Ooh, how about that one? That's a sweet one there. How about that one? Do I see that one?
I think so.
It might look like a pyramid, right? Here's another one, that's not bad. So this is more the standard way you see them. There's another one there, and there's one there. There's a lot of them in here, but these are pretty good. That one's not quite so good, right? That one's okay, not good, right? There's a lot of them in there. Pyramidal cell layer is fairly large, by the way, relatively. So if we're looking at this layer here, right, you've got your molecular layer, your outer granular layer, which is right about that big, and then your pyramidal cell layer goes like down to here. It's a pretty good sized layer. All right, what's next? Then we go to part 2, right? This one. Which has your inner granular layer, your ganglionic layer, and your multiform cell layer, right? Those are the other 3. It's when you get to those inner 3 layers that things start getting messy. All right. One of the things that's a little helpful here, and this one actually shows it pretty well, this little area here, is what are you seeing if you kind of, again, step back and look at this a little bit. What are you seeing, like, what's this? What are those things? They're almost like little what? Like little rivers or something, right? Little fibers running through this, right? Does this look anything like the image that the guy showed on the TED, by the way? Not really, right? And why is that? What magnification was he looking at? Probably a little higher than this, right? Maybe 200 times more than this or more, right? This magnification is what? 100. And he was probably looking at 3,500 or something. So if you looked at that high mag, you'd see something different too, right? So that up there on that edge is pyramidal cell layer. This is medulla here. This is the edge of medulla here. So between here and up there, you have 3 layers. Everybody see them? What do you mean no? They're easy to see. What are the names of them again? So the first layer is the outer 3 are what again? Molecular, outer granular, and pyramidal, right? And then the inner 3, starting from the pyramidal cell layer, is the inner granular, the ganglionic, and the multiform. So the multiform is going to be closest to this, right? So this at least we know is multiform, right? What's multiform mean? Literally, what's it mean? Multiple different shapes, right? So you got all kinds of different shapes of cells down here. Grainy layer, where's the inner grainy layer? Do you see a grainy layer there? Maybe, not really. You want me to find a better area? I can look for more. Let's see, hold on. That's not bad, let's look there. Maybe. This is probably the best spot we got. This is really nice, how about that? That's nice, I like that. So on this image, okay. Medulla here, ends there, right? Pyramidal cell layer, you can see a pyramidal cell there and a pyramidal cell there, so the pyramidal cell layer goes to about there. So between here and here, you got 3 layers. Yeah.
Could you maybe point us out to the medulla on the handout?
Can I do what?
Point out the medulla on the handout.
This handout?
Yes.
It's not on that picture.
Is it on the next picture?
This is a good slide for pyramidal cells because it's in the pyramidal cell layer. That's all that you're seeing, right?
On the back as well.
This one molecular layer out there, right?
Okay, so one to do was like the top.
And
Yeah, top right.
that's probably the multiform layer there, so it goes from there to there across the board. Does that make sense? This one has them all, it's really low mag though, right? That help at all?
Yes, it did.
At least giving you reference to what the slides are? Those are all the same piece of tissue, by the way. Just different magnification, different areas, right? Where's that one with all the lines in it? Here you go. This has got the layers in it too, right?
Yeah, wasn't that?
That's what those weird bars are, right?
Wasn't that the first
Showing the
three
different layers.
then?
It's got the molecular layer, the outer granular, and the pyramidal cell layer. That's it, right?
Right.
Okay, back to this. No?
Keep going, you're doing great.
You keeping up? All right. So, what did I say this was? Here?
The medulla.
Medulla. So what's this layer then?
Multiform layer.
Multiform layer, where you have multiple different shapes of nuclei, right? And it ends right about there, right? What do you think this layer is here to about, from about here to about there? What's the next one up, right? What's it called?
Fusiform.
I don't know, where's my list? So there's intergranular, something, and then multiform, right? What's the one in between? What's it called? What's the name? Oh, I don't know, can't find it. Where's my note? I lost my page, you got me all messed up on this.
Oh, ganglion.
Ganglionic, what's a ganglion? What's-
Bundle of cells, a bundle of nerve cells. All-
True, but what, ganglion cells, like in the dorsal root ganglia, what do the cells look like? A ganglionic cell is a descriptive term, I don't know if you know that. Kind of like I said that a vesiculated nucleus is a very specific structural term, right? Ganglionic cells are basically round in shape. So what do you think the cells are gonna look like? Round. Like these cells all look round in this area right here. The intergranular layer is a weird little transition zone between the pyramidal cell layer and the ganglionic layer. It sometimes shows up well, sometimes you never look at it, you never see it. It's one of those weird little layers that is hard to catch unless you get lucky, really. The outer one's always distinct. I don't know why that's the case, but that's always the case. So, 5 layers relatively reasonable to see, the 6th one's hard to see. Okay? So was today an easy day or a hard day?
Depends on.
I guess we'll see.
Like.
Well, on the second lab practical, the most missed questions are always these layers. Everybody always misses them, I don't know why. And I usually ask the easy parts too, right? Like I'll ask the pyramidal cell layer because look, there's a pyramidal cell, right? Or I'll ask like the molecular layer because, you know, it's smooth looking, right? What are we thinking? What time is it? I'm early, I got lots of time. What do you want to talk about? Want to talk about the coffee in my office?
Yes.
Coffee maker, snack bar. I have all kinds of stuff in my office. What?
Didn't we talk about this last week?
Have you not seen my stuff in there?
I haven't seen the snack bar, no.
In my conference room?
That's where I got those chips yesterday.
What?
That's where I got those chips yesterday.
I have chips, I have little bags of cookie things.
That's what we were getting that stuff from?
All stuff I can't eat, by the way.
Why do you have it? Is that not a temptation for you? Just like, ooh,
It's a perk for my faculty and any students who know about it.
gotcha. Well, I appreciate you for sharing.
I drink the coffee, the tea, and that's about it. I don't eat much else there. Anyway,
This does.
for those 5 people in the classroom who've actually been in my office. Speak.
If we have extra time, can you remind me what the difference between fibrous astrocytes and the protoplasmic astrocytes are?
Right. Protoplasmic literally means like blob shaped or ball shaped.
Gotcha.
Fibrous means long and stringy, right? And when we looked at those, which slide is that? Was in this one, right? So let's look here. Let's look here at the sweet vine. So for example, this is as high as I could go without going to oil. Okay? First, let's look at, this is cerebellum, right? How about this guy right here? If you focus in and out on it a little bit, you can see it's got extensions. You can see that, right? See the little extensions there? And there's the main part of it there, right? So are those long stringy extensions or short stubby extensions? Short and stubby, that's a protoplasmic astrocyte, right? Let's move to a different part and look at the longer ones here. Let's go up here and look at, I don't know, pick one. Where's a good one? Any one of these is good. They all look kind of weird. Pick one, I don't care. How about that one? There's the main part of it there, and these are the extensions from it, right? Are those short and stumpy or long and stringy? Long and stringy. Fibrous astrocyte, right? Pretty straightforward. Standard situation is that the fibrous astrocytes are in the cortex, protoplasmic astrocytes are in the medulla.
Is there a structural reason for I get the fibrous astrocytes.
Remember, astrocytes are for holding it together, right?
Right.
And remember also where the myelin is and where the myelin isn't.
Gotcha.
Yeah.
Makes sense.
Is there an example of microglia on the last slide of the
Microglia?
Yeah.
What about microglia?
It's just one of the things that my book here's forgiven.
Okay, so let me show you microglia on this one. Not that you'll get it, right? So for example,
Another like really hard to find.
there's a microglia there, not that you would have realized that without me telling you. Everybody see it? Right there. See that little string looking thing right there? That's a microglia. That's about what they look like. It's just a little disconnected little string of something, right? And they can be any length, they can be any density, they can be contracted and be shorter. There's a lot of variation in them, like macrophages, right? So here's one here. Look at this one's all bunched together. That one, right? Other than just a little piece of dark material, that's what I can tell you, right? They are the immune system of the brain. They're not very good at their job. Infections in the central nervous system are very bad, right? And what's it called when you have an infection in the central nervous system?
Bad day.
Meningitis, yes. Alright, anything else you wanna hear while I'm here? You've had too much, your brain's full?
Where can we turn this live in?
Say that louder.
Where can we turn the live in to?
I'll put something on Blackboard today.
Thank you.
Remember, no lab next week, right?
Oh, yeah.
No lab next week, yeah.
Cause it's fall break.
Exactly.
And I'm not coming in on Thursday, fall break.
Test on Wednesday.
What's this weekend?
Homecoming.
Homecoming for all you people who care.
But I know.
I need to recharge my,
Oh, yeah. Every week.
very introverted,
Oh no, she's not taking it seriously.
I've seen everyone
Do you hunt anything? Hunt?
Hunt,
Deer?
like animals?
Well, not people. Or maybe, I'm not gonna judge you too hard.
I have been hunting, not recently in my world. I
Yeah.
do, feed the deer around my house and watch them, but I don't hunt them. If I went hunting today, I'd probably use a camera. The only real hunting I do these days is I do it at like Walmart, because it's easy to sneak up on them.
You're not a fan, or?
That's a whole nother story all by itself, right?
Fair enough.
I was in the Corps, right?
Gotcha. Fair enough.
Don't need it, right?
Sorry for asking then.
Is season opening up or something?
Yeah, bow
Bow
season
season.
open up first.
I knew it was coming. I forgot it was it's 1st of October, right?
Mm-hmm.
And gun season opens up around Thanksgiving, right? Is that right?
Yes. Mm-hmm.
I'm aware of it because the deer tend to scatter about that
Yeah.
time.
Yeah, that's fair.
How did your dogs do in the competition? You
Uh,
had 3 of them in, right?
I had 3 dogs in it. I had one dog that won Best of Winners one day, and that was the only thing I won.
That's not a bad.
It's not bad, it's
win,
fine,
though.
yeah. When there's only 10 winners dog given out the whole weekend, actually 5 winners dog given out the whole, there were 5 shows, so it's pretty good, yeah, not bad at all.
Nice.
I do need a break from dog shows, so, oof. This one was way too much work. Anyway,
Imagine running or being in it as well as like running it as well. That
well, I didn't run the whole thing, but I did a reasonable part of it.
yeah.
Did Jamie go show the dogs?
Jamie was there, she was showing somebody else's dog.
Oh, really?
Yeah, not mine.
All right. Well, have a great rest of your day.
All right, what am I gonna see, lecture tomorrow, right, lecture?
Yes.
Friday, tomorrow's Friday, yeah?
Tomorrow's Friday.
Okay, get my brain working.
I'll
Was
try and
not-
get mine too.
You 2 grad students did just fine on the test, even though neither one of you got a perfect score on it.
I know, I didn't.
So close though. So close.
I figured I
am curious.
They're just a couple of little minor things, nothing major.
Do you remember? Do you happen to
No, I
remember?
don't remember what they
Gotcha.
were.
Gotcha.
I know that one of you got a 98, one got like 95 or something, but neither one of you missed anything that was pertinent, just little minor picking things I picked out of them. The
That's
one that got
good.
me was the guy who got a 50 something.
That was kind of crazy.
Yeah,
How do you get a 50 something on a take home test?
I don't know. Yeah.
'Cause that's the part that bugs me, right? If you have a take home test and you don't get a really good score on it, what are you doing? I don't know, I don't get it. Doesn't strike me in my brain.
I don't know. I don't know.
I guess it's just not